just for completeness, I wanted to just share some quick bullets with the community. Again, just for completeness, there are some observational studies happening, things we didn't talk about specifically in this conference. There's an ongoing Type A remote natural history study, a Type C natural history study that actually, I believe, just got ethics approval. And so hopefully we'll be beginning to enroll soon at the clinical site in France. But there will be a remote component to that for other patients, and later a site in Texas. There's an ongoing Type D natural history study. They're not specifically accepting new patients at this time, but encouraged to reach out if you are someone with Type D so that the investigators can know you're around. Next slide. There's also the BOLD study, and this is directed by Dr. Dixon at Washington University, and this is looking across lysosomal diseases. Right now, they're particularly focusing on Sanfilippo type B. It's remote participation. There's another study going on at the Lysosomal Research Disease and Treatment Center in Fairfax, Virginia, and a study out of France, which is just enrolling French patients, and that's a 5-year long-term, ongoing study. Next slide. Some other studies related to treatment or long-term follow-up. You heard about the upcoming Epidiolex study from Jonathan Acevedo earlier. There's a study looking at Ambroxol, which is also not yet enrolling, but is anticipated to begin at the site in Fairfax, Virginia, for adult patients. Just to follow up on a previous gene therapy study that was conducted by Lysogene, that now is being managed by a nonprofit called Blue Daisy under the same directive. Karen Eisch is still leading Blue Daisy, and she is arranging for long-term follow-up of those patients so that we can really understand the full data set there. And the gene therapy study from Esteve does also continue to follow their patients. Again, to reach back to what Dr. Lau was saying, long-term follow-up is critically important. And I think in gene therapy studies, when you're not continuing to receive treatment, it can kind of go out of your mind as to how important that is. But the data you're contributing actually truly impacts whether these drugs get approved or not for everybody. So your contribution is vital in the long term. Check out our website for easy access to clinical trial information. And then always we would refer you back to the clinical trial websites or the investigators or companies themselves, but we're always happy to help get you to the right place as you need it. So now I'll introduce our last two speakers of the conference. We have joining us from England late in the evening. Thankful that Dr. Simon Jones can join us. He's a metabolic geneticist, medical director of the clinical research facility there at the University of Manchester. And he'll be talking about OTL-201 gene therapy for Sanfilippo type A. And then we will follow directly with a presentation by Dr. Anna Claire Meyer, who is a neurologist and medical director at Denali Therapeutics, talking about DNLI, I won't say all the words, but their type A enzyme replacement study and their work with the START program with the FDA. And then joining us for Q&A will be Kim Ramsey, who's the director of patient advocacy at Denali. Dr. Jones?
Hi there. Thanks very much, Cara. Thank you, everyone, for being there. Thank you for inviting me. It's a great meeting. I've enjoyed the last couple of talks an awful lot. So I'm Simon Jones. I'm a pediatrician from Manchester in the UK. I've been looking after Sanfilippo families here for 20 odd years, and I'm doing a number of trials, not always with the answers we wanted, but doing a number of trials over those 20 years in Sanfilippo, and trying to get closer and closer. So I'm going to talk a bit about our phase 1/2 study. Some of you may have heard some of this data before, because I presented a year ago and perhaps before as well on this study. So this is our phase 1, so the very first in human study for our lentiviral gene therapy study in Sanfilippo type A. This started off as a university type trial here in Manchester, and the drug, the gene therapy, has been licensed to the company called Orchard Therapeutics. Hence, it now has a fancy industry title. And so I'll talk about the data so far on our 5 early stage patients, and the now 3 or 4 years follow up we have for those children. Next slide, please. Thanks very much. So obviously everyone here knows about Sanfilippo. This, because it's a gene therapy, we replace the missing gene. This is disease or type, Sanfilippo type specific. So we started with MPS IIIA. And our idea was that we knew that bone marrow transplant in MPS I corrected that disease, or at least a large part of that disease. And that's the same pathway. It's still heparan sulfate that builds up in the brain. So it should help in MPS IIIA. But when people did do in the past bone marrow transplant for MPS IIIA, there really wasn't the same outcome and benefit, and there wasn't stabilization or lack of progression of neurological disease. And so the theory was that bone marrow transplant from an unrelated donor really didn't give enough enzyme to the brain in Sanfilippo to stop that progression. Whereas if we could do a slightly more fancy version of bone marrow transplant, so a gene-modified bone marrow transplant, and give even more enzyme, then maybe that could work. And so we've been working on this for about 18 years now. Heather was talking in the last presentation about how long some of these things take in industry. Well, probably if you're working on these things in a hospital and a university like we have been, they take even longer. So it's taken us 18 years to get from what felt like a really good idea to the end of our phase 1 trial. Next slide, please. So to give a bit of an idea of the concept of ex vivo stem cell gene therapy, sorry for the name of the study. It's just terrible, isn't it? It's very difficult to make this straightforward and simple, but essentially, instead of using blood stem cells from another patient, from another person as a donor, and relying on those stem cells leaking out or secreting the enzyme that's lacking in the patient who receives them, what we do is we take the blood stem cells from a child with Sanfilippo, and then in the lab, in a very clean, careful lab environment, we mix those cells with the viral vector, the lentivirus, which puts the missing gene, the SGSH gene, back into those stem cells, so that they're all corrected, and not just corrected, but actually we modify the gene so that those cells are going to make lots and lots of it, not just the sort of standard amount, but extra standard, lots and lots. And then we put those stem cells back into the child that they originally came from. So the child feels like, if you like, they've had a bone marrow transplant, but the cells they have are not from somebody else who's somehow better or not as ill, but actually they're their own cells back. So that's a safer bone marrow transplant, but it's not just safer, but it's better. And so there's an awful lot more enzyme made. I often say it's like turning up the volume. So instead of, those cells just making the standard amount of enzyme, you can actually make them make a vast amount of enzyme. And so that was the plan. That was our original idea, not just ours in Manchester, but a number of other centres. And we're very encouraged to see that this is now an approved treatment, this idea, with a different gene is now an approved treatment for a brain lysosomal disorder in the US and Europe called metachromatic leukodystrophy, or MLD. So we know this can work from other similar disorders, and we think it has a good chance of working in MPS IIIA. But these are long studies that take a long time, and are not straightforward. Next slide, please. So we took 5 patients on our phase 1 study, all who came to Manchester. We took really young patients because we knew we could only treat 5 initially in our study. We had to make sure that they gave us the most useful information. And so this is where trials are different from clinical medicine. We're not just trying to help all of the MPS IIIA kids that we have. What we're trying to do is pick those MPS IIIA kids who could help us answer the question in the best possible way. So these are trials, like all trials are selfish. They're aiming to answer the question rather than just to help everybody. And so we wanted to take kids who have not yet developed many of the brain features of MPS IIIA, so younger than 2 years of age, with good sort of essentially normal brain function, but who we knew because of their genetics or a family history were going to have rapidly progressive MPS IIIA. So we treat these kids before they show lots of signs of the disease and then follow them up for a long time. This is a really hard trial design for everybody, for funders, for the families, for clinical trial design, and for clinicians. It takes you treat really young kids, so it's hard to find these kids, and then you have to follow them up for a very long time before you really know whether the treatments worked or not. This is absolutely why Heather's description of the accelerated approval route is fully supported by us, and the way we need to go really for MPS III families and for MPS III therapies. And because this kind of trial design, it's almost impossible to do, actually. We've been very lucky to get through so far, I think. And we could have had much more difficulty at lots of points, and this whole program could have gone by the wayside, as many MPS III programs have, unfortunately, because of all of those challenges. So we went through our kids who went through this process were safe. They didn't have major adverse events. They didn't have complications of this gene therapy. The side effects and toxicity were that which was related to transplant itself. All of our children survived transplant and did well, and we're now following them all up. We're now over 3 years out. What we find when we measure the enzyme levels in these children, so leukocytes are the white blood cells. When we measure the white blood cells, we find that enzyme levels, the enzyme that when deficient causes Sanfilippo type A, SGSH, that this enzyme is at least 100 times the normal level, if not more. When we look at the plasma level, so that's the enzyme that those white blood cells have made and leaked out, so secreted, that can be then taken up by other cells in the body, including brain cells, then that's around about 10 to 15 times the normal level. And when we look in CSF, which is the spinal fluid, the fluid that Heather described that surrounds the brain, then we see that there's more than normal healthy person levels of this enzyme in the fluid that surrounds the brain, telling us that we got there. We got cells to make enzyme, that the cells secrete enzyme, and actually that process happens in the brain as well. So that's what we wanted to do in the study. Of course, what we really want to do is make kids better. How do we know that happens? Well, the next slide might show us this. we measured heparan sulfate. That's the MPS or GAG chemical that accumulates in Sanfilippo that we know causes the problems, causes the neurodegenerative disease. And what Heather was describing, we argued with the FDA that should be used as a primary endpoint and key biomarker in this disease. So we could see that heparan sulfate in all compartments of the body, so the blood, the urine, the spinal fluid, really everywhere we could measure the heparan sulfate, it was getting so low to the point of normalization that we couldn't tell whether the samples were from a Sanfilippo child or an otherwise healthy child. And we know that that's very different from standard bone marrow transplant, because colleagues of ours who tried bone marrow transplant in the past for kids with MPS IIIA, even when very early, they could not show measurable enzyme levels in those patients, nor could they see significant drops in CSF heparan sulfate. Next slide, please. This is a lot of fancy analysis, which simply tells us that we don't think we've done any harm. And what we've tried to do with this gene therapy is to alter the DNA of those blood cells. So we put that missing gene, the one that's not there or not working properly, in DNA in kids with Sanfilippo, put it back in again. But what we don't want to do is mess up your DNA and cause problems, specifically cancer. And so this is a whole pile of technical work that we do to try and make sure that we're not making kids at risk of cancer. And there's absolutely no evidence of that so far, but that clearly doesn't make us complacent about this. And we will keep monitoring this very carefully. Next slide, please. Yep. So when we make a kid make an enzyme that they previously have never made, because they have a genetic deficiency of it, it's possible that antibodies could happen. And so we watch for that very closely. What we see is that the bodies of our kids, the immune systems of our kids, recognize the enzyme that we put back in as different and something that they've not seen before. And so they do make some antibodies against it, not very high amounts, and those antibody levels come down very, very rapidly over the time period that we essentially see when other MPS kids have transplants. We need to keep monitoring this. However, we don't think they cause a major issue, because when we look at the CSF heparan sulfate data, we see that actually that does come down as much as we would have hoped for. But we need to watch this very carefully. Next slide, please. Okay, so what do we really want to do? Well, we don't treat the kids in our study or any of our kids to change an enzyme level or a heparan sulfate level. We try and improve lives. This is a phase 1 study, so the key endpoints, the primary endpoints, are all about safety and biochemistry and this sort of thing. But of course, we've been following up our kids over the long term to see what happens. And yes, this feels like a long term because we've had 3.5 years follow up. But of course, given that we treated some of these kids very young, under the age of a year, this isn't a long time. This is a disease that evolves over 5, 10, 15 years. And so what we're seeing is that 4 out of the 5 kids that we treated in the clinical trial have shown dramatic benefit in that they are developing almost within the normal range. So some absolutely normal and almost above normal, and some absolutely at the lower end of the normal range. So continuing to develop skills in a really impressive way that we don't see in otherwise untreated or unmodified children with MPS IIIA, at least the severe form. And indeed, we've been able to move beyond the BLE test, which has a ceiling or sort of top age limit of 42 months. And so when we've done a number of previous studies, we've not been able to get any MPS IIIA kids with the classical form of the disease beyond that test, because they reach the top of their development that they can manage, and they plateau and then deteriorate after that. So we think we've seen developmental gains that we've never seen before in our Sanfilippo kids. We think we've seen behavioral changes like tolerance of, as you can see from the photographs, hearing aids, glasses, we've seen continence, gain of speech in more than one language. We've seen these where we haven't before, but this is still early. It's still variable, so patient to patient variable, and it's still very early days. So we need to watch more, but we also need to treat more kids. Next slide, please. We're very, very encouraged so far that this therapy appears to be safe, insofar as the 5 kids we treated in the trial and 1 kid we treated outside of the trial. The therapy has done what we wanted in terms of the biochemistry, and we've done, which has done really everything we wanted, and doesn't appear to be toxic or unsafe at this point. But we will watch these kids very, very carefully. However, we do need to treat more children. Ideally, we would do that as part of a phase 3 trial. So that's as Heather and others have described, is a more complex trial with the primary aim to prove that this therapy works and clearly changes the underlying disease. So it's efficacy rather than safety study. But for that, we would need more patients and more time. This product, this gene therapy, is now owned by Orchard Therapeutics, rather than us in Manchester anymore. And so we are working with them and regulators to see how we could move towards, as soon as we possibly can, a phase 3 study, so we can start treating some more kids, because we haven't treated a kid in nearly 3 years now. So we're getting itchy. I'll stop here and thank the huge team we have in the hospital, in the university, and more widely in Orchard. We thank the team at Great Ormond Street who've done all of our viral transductions. This is a huge amount of work when you're not a pharma company, to do this as a clinical academic team. So thank you. And the support we've had from the MPS societies all around the world and the Sanfilippo organizations has been critical to this, to find patients. None of our 5 patients were from the UK, and we recruited all 5 patients during the worst of COVID lockdowns. And so there's no way we could have done that without the global community of Sanfilippo patients working together. So thanks a lot.
All right. Can everybody hear me? Gonna hope that's a yes. Wonderful. All right. Well, good afternoon, everyone. Thank you so much for staying to hear this talk. I feel like it's always a hard challenge when you're the last talk in a conference, and it's been really inspiring to see the sessions. So I think thank you to everyone who's here, and to the Sanfilippo Foundation for Cure Sanfilippo for putting this on, and really, I think everyone for sharing such, I think, personal and moving details about what families go through when a child is diagnosed with Sanfilippo. So next slide. I'm an employee of Denali Therapeutics, and I'll be sharing a little bit of information about our DNL126 program. I do want to note it's an investigational drug and is not approved by any health authority, and no conclusions about either safety or efficacy can be made. Next slide, please. So I'll try to go under the 15 minutes since I know it's been a long 2 days for everyone, but I want to share a little bit with all of you about Denali as a company. provide some information about our enzyme transport vehicle technology and DNL126, which is a therapy being developed for MPS IIIA, and tell you a little bit about our ongoing phase 1/2 clinical study. Next slide, please. So first, a little bit about Denali. This company was founded in 2015, really with the sole purpose of defeating neurodegeneration. We're based in South San Francisco and have sites in Salt Lake City and Zurich, Switzerland. What I personally love about working at Denali is a commitment that I feel is foundational and informs all the work we do, which is really to patients who are living with neurodegenerative disorders, including lysosomal storage disorders. And I think the partnership and contributions that we have both from families when we work to design a study, from advocacy groups like Cure Sanfilippo, and I think as Dr. Jones pointed out, really the whole global community of families and children living with lysosomal storage disorders. Without that, we really wouldn't go very far. And so I think we're so thankful, and we remain committed in our own partnership with families and caregivers and patients themselves to design our studies and to ensure that what we're working on really meets the needs that are most important. Next slide. So next I'll tell you a little bit about DNL126. This is probably our busiest slide. DNL126 is an enzyme replacement therapy. And so what that means is we are taking the SGSH enzyme, which is the enzyme that is either deficient or malformed in MPS IIIA, and we are replacing it. I think as you've heard in some earlier talks today, it can be challenging to get a big protein into the brain. And the reason for that is something called the blood-brain barrier, which is a very specialized filter. And its job is to let good things into the brain, good things being things like nutrients or iron, and to keep bad things out, infections or proteins that just shouldn't be there. And so the challenge with treating a disorder like MPS IIIA is getting a big bulky protein through the blood-brain barrier into the brain where it needs to get to have the most clinical benefit is quite challenging. And so, the SGSH enzyme is fused to a second protein called our transport vehicle, which is engineered to have a binding site to something called the transferrin receptor. The transferrin receptor is naturally occurring on the walls of all of our blood vessels, and its normal job is to bring iron from the blood into the brain and other tissues. And what the TV does, the SGSH enzyme, is really hitch a ride on the transferrin receptor to get the enzyme from the blood into the brain and other tissues in the body. I think the other thing many folks may be familiar with our work in Hunter syndrome or MPS II, where we have a phase 3 clinical program replacing the enzyme that is deficient in MPS II. And so, I think we're thankful to be able to build on Denali's experience, both in designing the molecule and running the trials. Next slide. So next I'll tell you a little bit about our ongoing study. I think as Dr. Jones pointed out, that the titles are kind of long and clunky. Go ahead and go to the next slide. But our phase 1/2 study is an open label study, which means both the patients, families, and investigators know what treatment the individual is receiving. And so all the participants in this study are receiving active treatment with DNL126. And the goal of this study is really to understand both the safety of this molecule. This is the first time this molecule is being given to human beings. And so we spend a lot of time making sure it's safe to give, and also to understand two things: the pharmacokinetics, which is how long the drug stays in the body, as well as the pharmacodynamics, which is measuring the effect of the drug in the body. So right now, the core study period is 25 weeks, and we continue to follow children up to a total of 96 weeks. We have around 20 participants who will be enrolling in 5 different cohorts. And if there are other questions kind of about the detailed inclusion and exclusion criteria for our study, that can be found on clinicaltrials.gov. And next slide. But I'll tell you a little bit about them. So the study is divided into 5 different cohorts. The first 3 cohorts are really to look at the effect of different doses of DNL126 and to assess the safety. And as we described, we kind of go very slowly, exposing a few patients, checking to make sure that the drug is safe and well tolerated before we start giving additional patients the medication. We recognize there's been tremendous interest in this study, and I think just really want to thank all of the patients and families, both that are participating in our study and those that were not able to participate in our study. We're really thankful for your interest, and also for the patients that are in the study, your commitment to participating in studies that are complex and very burdensome for both the patients and the families. We're so appreciative, and we really can't do it without you. And so I think for all of the participants, both potential and who are in our studies now, for your time and your generosity. Cohorts A1, A2, and A3 are currently have all the participants identified. We do still have cohort B1 and B2 available for enrollment. I think, as Dr. Jones said it really nicely, which is that studies are selfish and that our goal is really to generate the information we need. I think generating the information is maybe not the selfish part. It's really a critical step to make this medication available as quickly as possible to the broader community. However, the first cohorts are quite narrow because the goal is to really understand as quickly as possible what effects the medications are having on participants. So the current cohorts were focused on children who are less than 28 months old at screening, and children who have a severe phenotype or severe presentation of MPS IIIA. Because we recognize many young children with MPS IIIA may have an older sibling also that have MPS IIIA, our cohort B2 is to enable recruitment of siblings of our cohort B1 participants. And next slide. I think just to close out, one of the things we're really excited about for our program is that we were selected earlier this year to participate in a new program that FDA is piloting. The goal of this program, the START program, is to help accelerate development of new therapeutics. It allows us to work more collaboratively with the FDA and have sort of enhanced engagements with them to help speed development of our therapeutics. We're super excited to be a part of this, and I think announced earlier this year, and are grateful to Cure Sanfilippo also for sharing that information with the broader community. We're looking to build on efforts like the ones that were discussed earlier, like the Regan-Udall Foundation meeting, and really using biomarkers like heparan sulfate to speed development and really make sure that we can get all of these medications to patients as quickly as possible. And next slide. So I think just to close out one more time, just a tremendous thank you to all of our study participants, to the physicians and investigators that make our studies happen, to all of the families who were not able to participate in the studies. We're very hopeful that we can make those opportunities available in the future. And then I think just a last thank you to the Cure Sanfilippo Foundation for giving us the time, and for all of you who are listening for staying so long to hear what we have to share about our DNL126 program. So thank you.
Hello, and welcome, Kim, to the Q&A.
Hi, everyone.
Hi. Well, thank you guys for the excellent presentations. We have a couple of questions. Let's see. For Dr. Meyer, does Denali have any plans for a 3B program?
So I think we would love to work on all the MPS disorders. We don't have plans that we can share right now about MPS 3B, unfortunately, but I think should we have that in the future, we will certainly make sure to update Cure Sanfilippo and other advocacy communities to get that information out there.
Okay, good. And Dr. Jones, could you comment on what the length of follow-up is typically for gene therapies that are delivered through lentiviral means, and how that might differ from some of the AAV therapies?
Well, we should all be following up patients who've had a gene therapy for quite a long time, really, at least 15 years, because these are really quite new therapies. People have been, when I started, I'm going to betray my age maybe here, but when I started in Manchester in this area in MPS diseases 20 years ago, I was told, oh, gene therapies, they're just around the corner, it'll be fine. Everyone will be cured. And you know, things have taken a bit longer than we thought and have been a bit more complicated. And so we really are still trying to learn and understand some of the very basic things about gene therapy. So I think it's very important that patients are followed up for a very long period of time. I think, you know, you just announced actually in the bit before I talked about some of the companies who did prior gene therapy studies, really trying to find those kids again and get them back into a follow-up program, because from a safety point of view, as well as how well do these therapies work, it's super important. I'm a pediatrician, and I'm accused by my adult colleagues when I transition my kids who are then young adults into adult care that I have a very children-focused sort of viewpoint, and I think sort of life is a very short period of time. And really, you know, if we do hope to really make a really big difference into the lives of these kids, and we're talking about survival for 20, 30, 40, 50 years, then, you know, we need to make sure that actually people are healthy and well through that kind of length of time. And that means finding ways to follow people up way beyond traditional clinical trial ideas and setups, because this is important. You know, we need, you know, I want people to be well for decades. I used to think that, you know, having complications when you were 50 was a minor problem, but now that I'm 50, it feels very important. So follow-up, very important. The reality is most gene therapy studies have relatively limited follow-up. We're now starting to get, for some AAV trials and lenti trials, sort of 10 to 15 years follow-up. I mean, that's viewed as a major sort of achievement, and clearly is a long way from what families want to know. When I consent families for studies, including gene therapy studies, they say, well, okay, what's my kid going to be like when they're 20? What are they going to be like when they're 30? These are the right questions to be asking. And you know, we don't have clinical trials typically that run for more than 2 or 3 years. And so you see the discrepancy here. So these are really good questions, and keep asking them, and make us as clinical academic people and companies do the right studies that answer these questions.
And I think that kind of follows into then that need of, you know, when kids transition from pediatric care to adult care, how does someone who's been on a one-time supposedly durable treatment, how do, you know, those follow-up obligations transition over? It'll be another challenge.
Yeah, I mean, that's a whole terrifying. Yeah, we've certainly had so we've certainly transitioned a number of kids who've been on enzyme therapies and other types of study, and I think we've had to really work hard to bridge gaps and barriers between, like, even clinical and academic centers. You know, to say, right, we need to work together and find a way of identifying a kid that would, you know, a kid that whose parents considered them to be under me, and they're now an adult and they've considered themselves to be in a follow-up program in another center? How do we kind of make sure we know exactly who was who and who gives consent for what and all of this? It's a bit of a minefield, and it's not very straightforward, but it's very important, and so we have to get it right.
Yeah, absolutely. Denali team, whoever wants to answer this, do you have an idea, and I know, you're continuing to enroll cohorts, continuing to gather information, but do you have an idea when you might reach a point where you're able to share some initial safety or efficacy data? Like, what's the outlook for when that might be?
Anna Claire, I'll let you take this one. Start.
Yeah, and I realized I omitted to introduce Kim, who works in our advocacy team. And so I'm thankful for her to join and kind of help round out any questions that folks on the call may have. So I think, right now we try to present the data when it's sort of mature and we can understand and make sure that we have a full understanding of, the effects of our medication. So I think right now we're thinking about, when is a time we can first start sharing the information. We're excited to share it and we know folks are waiting for it. So I think we're working through that. I don't want to make any promises as to time, but we're hoping to at least provide, some preliminary information soon, and then, look forward to providing detailed information at an upcoming scientific meeting. And we hope we'll be invited back here as well.
Okay. Thank you. A question for you, Dr. Jones. You said you're itching to treat more patients. We would share that sentiment. And the question is, when will the phase 3 study begin? Any clues on?
No, I honestly can't. Thank you. I know, that's the golden question. I honestly don't know. It depends on a whole pile of things. We are moving from what was an academic study in Manchester to what will I hope and imagine be a multi-center international phase 3 study with a commercial vector and all of this sort of thing. So that depends on Orchard Therapeutics. It depends on the regulators and on the planets aligning and all of those things. So I can't honestly give a time scale. I wish it was yesterday. I can't give a time scale at this point because it would be unfair.
Sure. Understand. Okay. And Dr. Meyer, another question for you about patients in the cohorts and the different, centers or locations. And if the patients enrolled are spread evenly throughout the 4 locations, I'll just read it. When is it likely that the patients will be added that only qualify under the cohort 1 guidelines?
Yeah, I'll do my best to hopefully answer the question as it was written. I think for current patients, we have 4 sites that are active, and our investigators work really carefully, one, to make sure the sites are spread across the different sites, but also to, take into account patient and family needs and try to get folks to sites that are close and convenient for them. For cohort B1, which is the cohort that is currently enrolling, there, I think we're trying, because those patients are quite young and we know that participating in a study is hard, we're trying to match patients really to the site that's most convenient to them, and our investigators are working across the different sites to try to help facilitate that.
That makes sense. Then there's another question really about the START program, and if there's an opportunity for Denali to share the outcome of those discussions and the interactions related to the START program.
And there also, I don't want to make any promises that I can't keep with regard to timelines. I'm a stickler for that. Yeah, we're hoping to provide updates as our conversations evolve with the FDA. And we really work with the FDA for what can be shared about the kinds of interactions we're having with them. But we're super excited. I think we feel very honored to have been selected, and we're excited that MPS IIIA was selected as a priority for the FDA. So I think that to us, I think together with what Heather shared earlier about the commitment to accelerated approval, I think for us brings a lot of hope and excitement to being able to move our program forward, hopefully not with the 18 years that Dr. Jones has had to contend with. I think we're really hoping we can, put the accelerator on all of these programs to really help get, new treatments to patients faster.
Yeah. I mean, it is exciting to see, the attention that FDA is giving to our disease space. And I think, the learnings, hopefully, from going through those frequent interactions and the opportunities the START program provides will translate across various programs, hopefully. Dr. Jones, one last question, and that is your opinion on the safety with viral gene therapy for things like cancer. And I think, citing back to some older studies where maybe there were some incidences of that, but, perhaps we have different technology now. Maybe you could comment.
These are really important questions, and we're very open and transparent about this when we consent families and talk to families about this. This is it's clearly something we take seriously. We want to make kids better, not make kids worse. And so when you're, cancer's in some ways a genetic disease, although we don't think about it like that. It's a disease where when genes go wrong, when we mess with a child's genome, we run risks. And so some of the early gene therapy studies, by early I mean sort of 20 plus years ago, used viruses or vectors that were quite different to what we use now, and there were some very unfortunate outcomes with regards to cancer, essentially leukemias. So that was significant, of course, and really set the field back by more than 10 years. And we really had to go back to the drawing board, start again, work out what was happening. So now I try not to get into too much of the details. There are probably different risks for different types of gene therapy. And there are short-term risks and long-term risks. The short-term risks we see more in lentiviral type gene therapies, the type that I've been talking about. So whilst the vector that we use in Manchester for the MPS IIIA program, and it has been used in the MPS I clinical trial that's ongoing in multiple centers of the world, and indeed in the MLD program, MLD vector being licensed in the US and Europe, there's not been any hints or cases of cancer in using that type of vector. A slightly older version of that vector in a disease called childhood cerebral XALD we have seen some changes in some of those children quite early on that do resemble cancer. Most of those children, in fact, all of those children have been treated well. But that's a real sobering thing when it happens for everybody. We all take that very seriously. I work, as you can imagine, because there's a sort of a bone marrow transplant delivered therapy. I'm not a bone marrow transplant. I work very closely with my bone marrow transplant colleagues, who are also the ones who look after kids with leukemia. So they don't take this lightly. And we follow up all of our kids very closely. We think we have modified the vectors intensively over the last 20 years to do everything we can to reduce the risk of cancer, while still being an effective gene therapy. And getting that balance is something that has been challenging, in part because you try and do all of that experiment and in mice, because mice can have Sanfilippo too, you find out very quickly whether you win or lose, and it's not so critical whether mice win or lose as it is whether kids win or lose on all of that. And so, but what we see is that actually that's not as, some of these side effects don't come out in the mice and yet do come out in the kids. So that's why we have to proceed very carefully and very slowly. That's why it's so frustrating for everybody, for us, for you guys, for everybody, with gene therapies, and it's super frustrating. But I think so far there's been well over 100 children treated with our kind of lentiviral vector, not just with MPS IIIA, of course, multiple different disorders, and all of those kids look safe, and there's not a hint of leukemia in any of them, but we will look at that incredibly closely, and if there's a hint of anything, then we go back and screen everybody all over again. That's as good as I can do. It'd be lovely to say, oh no, there's nothing to worry about, but I'm not going to do that, because that's not where we are.
The truth is, I mean, people with Sanfilippo know what they're up against anyway. Yeah. So, but
absolutely
helpful to know that what we know now, right? We can make decisions on what we know now. Yeah,
exactly. And I often talk to the Sanfilippo kids that were consenting and saying, look, this is a very theoretical potential risk, but the reality is, 80% of kids with leukemia get better now and are completely cured. 30, 40 years ago, it looked as bad for those kids as it did for our kids with Sanfilippo. But that field has moved on. And so we need to learn lessons from that. And so, leukemia is something that actually we can deal with now. Actually, Sanfilippo is not. And so, we all weigh up those risks very openly and explicitly.
Absolutely. Well, thank you all so much for being here, for being part of the conference. It's been awesome. I appreciate the discussion. And we look forward to hearing more down the road.
Thank you so much, Cara. Thanks, everyone.